Okay, Luke. So he shows up with this thing in his head, and I'm like, you kiss ass, really? And then he puts on his screen and he starts showing us what it is that he has there. Luke, you are on mute.
I'm unmuted. I had to get my beer.
Go ahead. Okay, let's.
I'm at the other side of the pond.
Please put on your screen again and tell us what it is you're working on there.
Yeah, this is a water-based head cap, but I have to plug in the patient ground again. Where is it?
Okay, it was working perfectly, but he needed a drink that badly that
yes.
the entire thing.
Okay, I guess you're familiar with this.
Well, I certainly am. Yeah.
Yeah. So you can see the blinking of the eyes in the top channels because at the front FP, and I tried to show some alpha waves in the back channels, but
you're a little excited. Close your eyes.
I'm doing.
Yeah. How are these waves changing now that you have the drink in your hand and taking sips?
That's what I want to know.
Also, I put my hand on the. You can see more 50 hertz coming in if I put my hand on the transformator.
Yeah, that's in America.
Yeah, 60 hertz in Europe. Sorry.
Yeah, for 15 in Europe, 60 in America. That's right.
Yeah. No, I won't. I stopped sharing. It's not interesting.
It was to me.
It's fun. It's fun.
But,
no, I don't. Go ahead.
The last week I've read about everything about Mr. Lowe, so and it was very interesting. And also read his PhDs. Yeah, I have read your
call
PhD.
Me Dr. Lowe otherwise.
Dr. Lowe is also good. I also saw that one of your how do you say that? The people literatuurlijst. Your literature list. I saw Dr. Kemp and Dr. Kamphuis with a reference, and those are the people I did my master thesis at the Sleep Center in The Hague.
Dr. Kemp, you said?
Yep, Bob Kemp. You know him?
Not personally. I know some people in Liège, though.
Okay.
Yeah. Let's see. When I wrote the Cambridge Declaration on Consciousness, there was a professor from Liège who was invited to the conference, and he specialized mostly in comatose patients.
Okay.
Yeah. It's a wonderful center there. So you did your advanced degree there?
Yeah. No, it's not advanced. It's a master's degree.
That's advanced.
Right. I suspect we only have an hour of your time. As much as I'd like to introduce the folks, so I have a paid subscription and membership, and I like to invite all of my subscribers on in the morning, and we just say good morning and what's going on. The guy walking back and forth, Jonathan, there has been exceptionally busy. Jonathan, why don't you unmute yourself and tell Dr. Lowe what you've been working on?
Well, hi, pleasure to meet you. And we did a lot of work in Liège for Cephaly, which is a neuromodulator against migraines. Mm-hmm. But right now we're doing diagnostic testing. So we're bringing testing to the home through rapid antigen tests. And we've been busy for the past 2 years, as everyone can imagine.
Yep, wonderful.
Well, he modestly forgot to mention that he's like the number 1, 2, or 3 seller on all of Amazon's health section, and that he can't make the stuff fast enough.
Fantastic.
We're very fortunate. We have a really good product experience. USA Today gave us a top score, and we have 4.8 on App Store, 4.5 I think on Google Play Store, and quite a few other high reviews. So we're very happy with the progress. You have
a CE and mark and FDA?
We have EUA, so we have emergency use authorization, and of course the long play is disrupting the diagnostic world, bringing people access to the diagnostic information, creating better wellness and better decisions in the hands of individuals, rather than having to go out or getting that information faster, so that when you do see the doctor, you're seeing the doctor with mutual information rather than waiting for a post-visit experience.
Fantastic.
And he's also offered to send enough product so that everyone who comes to 10X can be tested, and we can be in a safe environment. And we can ostracize Eddie, for example, if he shows up from he lands and becomes infected. Is there anyone who wants to say anything before I put you all in the peanut gallery?
Just one thing, with respect to Luc's comment, the gentleman in Liège was Professor Stephen Lawrence. I don't know if you know him, but he's an expert in coma. Anyways.
Luke, you're on mute. If you wanted to say anything.
Oh, I didn't want anything to say. No, it's not only coma, but it's also about locked-in patients.
Yeah, that's right.
Yeah. So they don't move at all. They can move anything.
So
You put a
so you
head cap
know him.
on. You measure EEG, and then you see that they're still that they're locked in.
So you know Steve?
No, not
Okay,
personally.
Great guy.
I'm sorry to peel people off, but Greg, I don't want to make an exception because we have a special guest. Greg is a brand new member, and I'd love to give them a chance to say hi, unmute yourself, and tell us what you do.
Hi, yes, I run a small little medical component company called RAM Technologies. We provide PC power supplies for medical devices, and they're already 61 approved at 3.1 right now. So if you're looking for anything for your devices that run on a PC that need a medical, that need a power supply to already drop in, we got one for you.
Okay. Well, I've taken Andre off already, but I suspect that will be interesting for him. And John Tugwell, you wrote something in the comments?
Yeah, one of the companies I'm working with in the hearing health is a company called NeuroMod Devices, which is addressing a bigger issue than hearing impairment, which is tinnitus. And it's already approved in EU. We're actually going through FDA approval. I've introduced them to a larger company that may be interested in purchasing their company.
John, is that company in Ireland?
Yes.
I've been to their office next to the Guinness factory.
Okay, that's it. You know Ross then?
I do. Yep.
Yes. Yeah. Ross and I have been talking quite some while.
Let me understand, were you at the Guinness factory and Ross came over, or you were there?
I went to visit the company when they were NAMSA. When I was with NAMSA, we did a lot of their biocompatibility work.
Oh, that's right. Yes.
Yeah. So anyways, I was there in 2017 or something like that.
Yeah.
Yeah, we went to the Guinness factory afterwards. So
yeah, smart thinking.
I see. That does make sense. Okay, well, thanks everybody. And if you have anything you want to contribute, of course, as usual, just raise your hand or leave a comment. And so are you going with the sunglasses for the whole presentation, or do we get to see the whites of your eye?
It's a little bright here, but it'll get a little darker probably in a couple of minutes, so I'll take them off.
David Hampton, are you working on anything in Philip's space at all? I know you have your fingers in many pies.
Yeah, no, we're working on a cardiac pressure monitor that's going to be implanted. So lots of good things going on just in our internal audit. I had a BSI today, so I've been busy with QMS documents all day.
Wow. Okay. Well, thanks for making time. All right.
You over QMS. Happy to be here, Joe.
Yeah. Well, it's a tough choice, but business goes on. Okay. And then it was just two of us.
I love the print in the back, un dimanche après-midi de la Grande Jatte.
I have a story about that.
Me too.
I went to NYU undergrad. And back then, going to NYU was, it wasn't as big a deal as it seems to be today. My son applied, and it's like not the experience I had when I applied. And I got into a program called the New York University Scholars. It was an attempt to lure people who would otherwise go to Ivy League schools. Now, the fact that I didn't get into Ivy League is beside the point. Anyhow, so one of the benefits was a very robust cultural development program where basically we could go to theater for $5-10.
Fantastic.
The very first musical I ever saw was Sondheim Sunday in the Park with George. And it really stuck with me. And I've been a performer basically my whole life. I sing in a chorus now, and it was so fulfilling. It still stands as one of the best musicals I think ever, and it's certainly the most meaningful to me. So that's why that's up there.
The original is the Art Institute in Chicago.
Chicago, right?
And I had been hitting my head against the wall, trying to come up with a mathematical formalism to distinguish brain patterns. And I took a sanity break at the Art Institute, and I stared at this painting, and I realized if I could just turn brainwaves into little dots and obtain brain states based on the concentration of these dots, then I would solve the problem. And so staring at this painting actually completely changed my graduate career, and my PhD thesis was, a bunch of dots showing all these different brainwaves, which were mathematically separable. So that painting is very dear to me.
Ladies and gentlemen, the reason I have Dr. Lawan is, as you've already figured out, he's fascinating. And he's even willing to talk at an 8th grade level so that we have any clue what he's talking about. Seriously. I mean, I'm sure it would be very, very difficult for me to keep up with you if you were in a room of your peers. So with all of that flattery, I will conclude with, I meant to shave completely for you today, and my razor ran out as I got to here. My wife was like, well, go with the goatee. And I was like, okay, well, that's not really.
No, this is very GQ. You should keep that. It's good.
You think so?
Yeah.
It's hard to keep it at this length though, but I'm already doing this. And guys know that after COVID, I grew a full one. That was my COVID goatee. And I couldn't stop doing this the whole time. And I looked at videos and I was like, hey, your hand. And I'd be like, nope. And then I'd be like this again. So I don't know. Anyhow, welcome. And I don't know what's been going on for the last 2-3 years since you and I had a good conversation.
A lot. And I appreciate the friendship and what you do for the space, and your time this morning, along with the very, very informed and erudite members of your group. Thank you so much for taking the time this morning. It's a privilege.
I'm more than grateful for yours.
Thank you. So a lot has been happening. So I don't know if you heard, but there was a little pandemic that's been sweeping the world called COVID. And
COVID-19, right?
Yeah, I think so. I think it's 19. Yeah. And actually, as hellish as it's been for the entire world, there are a number of lessons that people had to learn the hard way. And one of them is that diagnostics are extremely important. And so what we're seeing in our space is a much greater appreciation for diagnostics, even though this is neuro and not, virology. And what we've also seen is there's been an appetite on the part of people who held real estate to do a number of deals in ways that they hadn't before. So what happened starting 2 years ago is it was always a mission of mine to help fellow entrepreneurs, especially scientists, engineers, and clinicians, obtain the tools necessary in order to, create a company and not be devoured by people who may have sort of short-term financial interests. So, we call this the Manhattan Project for the Brain. The USPTO actually gave us allowed us to use that moniker. So we started closing a number of deals. We did a deal with an accelerator in San Diego, then with a clinical trial facility in Wyoming. Wilson Sonsini, the elite, Silicon Valley law firm, put 10 times more in us than, a local fruit company and search engine company called Apple and Google. The order came straight from Larry Sonsini, who is personal attorney to Steve Jobs and co-founder of the firm. Kilpatrick Townsend, an elite law firm, also joined, and they specialize in intellectual property. Then we struck an alliance with the biggest international supplier of software talent, Silicon Valley. That's the University of Waterloo. And now we're in discussions with Johns Hopkins, as well as UCSD, UCSF, and MIT and Stanford, where I held positions for many years. So it's quite exciting because this enables us to put together an arsenal of resources, not only for NeuroVigil, but also for fellow inventors. And that means that we would be able to provide them, for example, with access to space, to legal support, to clinical testing facilities, without necessarily taking a piece of equity from their companies. So this is, something that will enable them to hit a number of milestones before they go for institutional support. That's one thing that's been going on. Another thing that's been going on is we're working now on the iBrain 3W, which is the smallest non-invasive brain monitor ever built. And the cool thing about this specific device is that it will be the same device that will be deployed to enable people with ALS and stroke communicate, not only communicate, but also play games. And we actually had a demo as early as 2013 with an ALS patient being able to communicate just with his mind. But the form factor is much smaller now. We will also use this technology for neurodiagnostics, especially brain cancer and Parkinson's. We're interested in finding biomarkers in asymptomatic individuals. And we are also going to go after the senior care market to make sure that to monitor individuals who might be over-medicated. Now, that's something we've done in pharma trials with Roche and with Novartis, collecting this kind of data. But this is, again, with a smaller footprint and in senior care homes all across North America. So we're very excited about this technology.
To even process how much you're even saying, I'm wondering, do you have, I'm a very visual person, is there a site I can bring up for folks to visualize some of the things you're talking about?
Sure. If you go to my website, I think
the NeuroVigil site?
Yeah, NeuroVigil.com. There should be a photo of me wearing one of the prototypes. There, if you go to leadership, you'll see a photo of myself wearing one of these small devices as a prototype.
About us?
Yep.
Permission?
Yeah, leadership.
Here?
Yep. There you go. That's the device. Well, that's a prototype of the device. So, it's tiny, and it's going to get even smaller. And the lovely thing is, we can collect all these biomarkers from people remotely. We have apps for Android and for iOS, and we are very excited in being able to get biomarkers from people who are asymptomatic. So, for example, if you look at Parkinson's, so we know that individuals who have RBD, REM behavior disorder, are extremely likely to develop Parkinson's or an alpha-synuclein pathology within 15 years. RBD is the inability to lose muscle tone during REM sleep. And it turns out that I happen to have, the patent on the distinction between REM and wakefulness on a single channel of the EEG. I did this for my PhD. I started doing this during my PhD. And what's amazing is that if you have Parkinson's or an alpha-synuclein pathology, the likelihood that you have RBD is about 1 in 3. On the other hand, if you have RBD, the likelihood that you will develop, Parkinson's or an alpha-synuclein pathology within 15 years is nearly 100%. So this is an extremely, extremely, extremely specific biomarker, and it has about twice the sensitivity of the most sensitive, Park and ping genes. So, this is something that we could do in the asymptomatic population. When it comes to brain cancer, we are interested in looking at changes in coherence within the brain and seeing if this pairs with changes in brain pressure. We're working with an inventor who thinks that he has technology to measure brain pressure non-invasively, as well as with Stanford University. So this is more of a moonshot, this specific project, but it would be certainly very exciting if we could find out if people are at risk of developing brain cancer and get them the help that they need. For individuals who are paralyzed, we have the Stephen Hawking Project, which started, with Stephen over a decade ago. And as early as 2013, we already had a patient who spelled the word communicate just using his mind. So we've been making this technology much faster now, and we will also enable individuals to play games. Again, there's no hole in your head. This is completely non-invasive. So we're excited to do all of this with the same device with a small footprint. And then the other big news is that we are really bringing the type of talent that I think has hardly been seen in a private company before. So over the last 15 years, I ran the company in a very esoteric way, in the sense that I refused, large capital injections from VCs. I bootstrapped it. I went to the market with Roche and then with Novartis. And I spent, I think, less than 6 million of the investment in 15 years. So we've been quite frugal for a biotech company. Now I'm recruiting some wonderful talent. So, Dr. Gabriel Vargas was, head of neuroscience biomarkers at Roche, and then head of neuroscience early development at Amgen. He's my chief medical officer. Noam Ziv was, VP of technology at Qualcomm. He's now my chief technology officer. Devin Goodman was head of technology operations for NVIDIA's presidential campaign. He's my chief operations officer. Dejan Vucinic, who was trained at MIT, at Yale, at Stanford, at Salk. He is doing R&D with us. Hans Gertz was an economist at Novartis. He specialized in pricing new technologies as they go into the market. He's going to be doing that kind of work with us as well. Crystal Dilworth is a Caltech trained neuroscientist who's known as Dr. Brain on CBS. She's a science communicator. She's our VP of communications, et cetera, et cetera, et cetera. We have a lot of Stanford faculty now moonlighting at the company, including one of the fathers of neuromorphic engineering and an expert also on point of care diagnostics. So it's a very, very exciting time where we're sort of coming out of stealth and bringing some wonderful.
I suspect this is not the Dr. Brain to whom you're referring.
Let's see, Crystal Dilworth, Dilworth.
DIR?
DIL, Dilworth. Dilworth. No, there's no R.
Okay. Here.
No, that's and it's not Brian, it's Brain. BRAIN.
That'll do it.
Yeah. Crystal, CRYSTAL. Crystal Dilworth, you got it here. Okay.
Yeah, okay. So basically what I did learn is that Crystal needs a little SEO work, but I'm sure she's got enough going on.
Sure.
But here we go. Molecular neurobiology. That sounds right.
Yep. Yeah. So she's known as Dr. Brain on CBS. They've got a show there, and she's an international science communicator. So she's working with us on communications. A lot of different people are joining the team now. So it's an exciting time. We're building a Bell Labs for the brain with people who are magnificently trained, and it's fun to see everybody being united by the same mission.
Let me have a sense, because you're such a force, and I'm not just being flattering. That's the way I think of you. Do you have full-time employees? Yes.
So the way I was running the company until now, was not with employees, but with contractors. Yeah. Many of them full time, but the point is, people get paid and they get their options and so on based on performance. And I had, when I started, full time employees, and I found out that if I converted people to contractors, that things got done much faster, yeah. and that people were happier. And instead of me having to run after individuals, they would run after me to show me the milestones that they had hit. So in terms of management and progress, going from employees to contractors was very, very helpful. What we're doing now,
perhaps my question really is,
yeah,
how many people are involved in, if you were to aggregate everyone who's touching one of these massive assignments of yours?
Right. Well, we've had about 80 contractors, at different points. In terms of this management team coming in, there are about 15 people. And what we're going to be doing is, for their first year, they have some very, very stringent milestones. And then based on their performance, then they'll be converted. The contract extends at least 4 years. So, and then there'll be full-time employees with all the bells and whistles that associate with that. But I'm a big believer in measuring performance rather than just hiring everybody as an employee at first. And that's been one.
I'm used to many more comments. I think people are stunned with the flood of information you've already shared that they don't know where to begin. I'll encourage you to
one about a necklace.
Write a question if you have something or a contribution to fill up.
There was one about. Nice to see Silicon Valley people working for the company. I heard. Yeah. That's actually, that's an interesting. And somebody noticed the chemical structure of serotonin. That's interesting in the Crystal's necklace. Anyways, so one of the gambles that's taken was coming to Silicon Valley. In fact, you and I met just after I had moved from San Diego to Silicon Valley.
I remember it was an what happened was I don't remember who was going to be my speaker that year, but they had to cancel last minute.
Right.
And I asked our mutual friend, Daniel Kraft, can you help me out?
Guy.
He put us in touch. And I think you basically made a flight round trip to spend 2 hours with us and flew. It
Yep.
was extraordinary. You saved me. So thank you.
It was a lot of fun. Thank you. But basically, I had just moved to Silicon Valley. I had been living in San Diego for about 14 and a half years prior. I had done my PhD there. I studied my company there. And, there were, it was an interesting situation. My office in San Diego was right by the ocean. It was beautiful. And I woke up one morning in my office, I showers in the office and so on.
Yeah.
And I went next door to that, Hotel La Valencia, very nice, iconic San Diego, institution. And I opened the menu, and my name is on it, the Dr. Low Vegan Delight. So I eat myself, and then I look at the paper, and on the right side, I see a lady I dated a couple of years ago. On the left side, I see a lady I just dated, and I thought, I need to leave this place. I need to leave right away because one can get used to this. I have no interest in being a big fish in a small pond. I was very, very lucky that I'd come out of some of the best labs, in the world, which happened to be located there. I had the top investor in town, backing me, but things had become too easy. And I felt that wasn't healthy for me or for my work. So I rented a room in a house in Silicon Valley. A Ukrainian friend let me rent a room. And I started, here, hungrier, and that was important, and then I recruited people here, and then I realized that there's this magic that happens in Silicon Valley and in very few other places in the world, where you find people who have worked in the places I've mentioned, Qualcomm, Novartis, Roche, Amgen, etc., and they're excited about the future just as much as you are, and they're willing to sort of leave their cushy jobs and come with you in the trenches and build something new, and that's wonderful. It took a while to get to that kind of critical mass, but I think that the gambit has paid off.
I would say. Do you make it down to San Diego often? Our keynote this year is Dr. David Albert. He's the founder.
Yeah, sure.
Are you friendly with him?
LifeCore, right? Yeah,
LifeCore.
Yeah, we've had a couple of emails. I don't know him personally, but we had a couple of emails a few years ago. It seems like
and as long the off chance that you're around, it's May 10. He'll be speaking, and if you're able to make it down there, believe me, I'll make room on the agenda for you.
Well, you're very kind. Sure. I have engineers in San Diego. I have a number of hardware engineers there, and my CTO is between San Diego and Silicon Valley. So I certainly do have many reasons to go back, of course.
Okay, well, I'm gonna follow up after this call to see if I can have you meet my friends in person. John writes, I just started working with a company called Biological Input-Output Systems with Joshua Miller, that you likely may know.
I don't know that company. I'm happy to learn more about it.
Well, I will put Mr. Tugwell back on, if for no other reason than I enjoy his accent. He also has good things to say, but I do enjoy his accent. Has it gotten a little less bright so we can?
Sure.
There we go.
Still a little bright. Okay.
A little bit.
No accent. I
Tell
don't have an accent.
Right. It's all of us. Go on.
It's all of you. Yeah, of course. No, they're working with the University of Utah Biological Input-Output Systems on a interface between the nerves in the bone and driving robotic exoskeletons, which is mostly my background.
Oh, yeah. Sure. Very interesting.
Exobionics.
I know Exo. Yeah.
Yes.
Yeah.
Yeah, for a number of years. So, and I'm also working with a spin-off of Exobionics, Rome Robotics.
So what happened to Exo? Because there were Berkeley Bionics, then there were Exobionics. I met two of the CEOs. Then there was also a bionics company in New Zealand that was doing some interesting work. There was Rex, exactly. So Berkeley Bionics became Exo, and then what happened to Exo?
Exo is still moving along slowly. Most of their income is actually from non-electronic driven devices. In other words, passive devices. The spin-off from that, Rome Robotics, Tim Swift, is got his own company in San Francisco area, and Nate, who was with,
He was a CEO. Yeah.
Yes, yeah. He's also CEO of a new robotics company that's actually doing what they call the eyebrow weaving, called Loom, L-U-U-M.
Oh, that's true. That's true. That's true. I saw that. Yeah.
Yes, yeah. So I'm still associated with this guys. This newer company, BIOS, is looking at driving. That's it. Yes, I spent a lot of time, several years with that device.
I can't remember, his name escapes me at the moment, but the former CEO of Exo is a friend of Daniel's as well. I met him at FutureMed some years ago.
Nate, is that Nate?
No, it's not Nate. Before him.
Yeah, there was one before him. That's
Unique.
Right.
He's CEO.
Do you remember his
of
name?
Unique.
And they used to have Oscar Pistorius as a
really?
spokesperson.
Wow.
Yeah.
That was pre-murders.
Yeah.
Murder is
Anyway,
really a career.
Interesting area, this whole area, but they want to drive, this BIOS wants to drive directly either prosthetics or exoskeletons.
Yeah,
Well, we're very interested in exoskeletons. The way we've been going about helping people who are paralyzed with their communication needs is to imagine that they could move a limb, and this started in Stephen Hawking's kitchen, where I would ask him to imagine that he could squeeze his right hand, his left hand, his right foot, his left foot, etc. And then lo and behold, we actually were able to detect that very often, and then also in trials in Philadelphia. So we would be very interested in controlling an exoskeleton with those biomarkers as well. That's later down the road. Right now, we're very focused on communications.
Great, good area. Thank you.
Thank you.
Pleasure. I like to be able to break things down to a level my mother-in-law can understand, and I think we could get there. If I were to call her after this and say, "Mom, you wouldn't believe who I had as my guest today," and I were to attempt to encapsulate what you do, what would be the best way to give her something tangible so that I could say, in 5 years, it's likely that, is there going to be something near in that everyday people could be like, "Oh my God, that guy, the guy who invented the."
Well, I hope I won't be personally known because I'm a pretty private person, but I don't mind for the work to be known, like the Cambridge Declaration, for example, that became world known, and I'm happy to be in the background. The idea is we're building a world where billions of people eventually are going to be able to access their brain in real time, non-invasively, and from home. And the reason that's important is because your brain has, nearly 100 billion neurons, and there's a lot of redundancy there. So, in order for you to develop a neurological disorder, most of the time, unfortunately, it means that many redundant systems and backup systems have failed. And by the time you wake up in the morning and one day you don't see anything in your left hemifield, for example, it means that mostly irreversible damage has occurred. What we want to do is to collect telltale signals that the brain produces non-invasively as this degradation occurs, in order to maximize chances of intervention, of successful intervention and recovery. So
what I'm hearing you say is your device is, I don't know if strictly is the right word, or predominantly diagnostic in nature.
Exactly, but we're early diagnostics.
I mean, if I can relate it to something every day that most people on the call can relate to, is a Holter monitor measures my, cardiovascular system for a period of time, and I wear it when it's prescribed to me for a period of 3 days to be able to get some reading.
Right.
Is that the kind of thing that I will go to my neurologist and I would be prescribed your device to see how I'm doing? Because I wouldn't wear it every day.
What you could envision is, so you could go to your mother-in-law and you could say, your doctor has just prescribed, a new drug regimen for you. And, I'd like to know if you fall under a, if you're responding the way other people are, or if you might have some side effects. I'll give you two choices. You can wear a non-invasive device built by a, Stanford MIT Salk neuroscientist who's been in the field for over 2 decades, or I can get a robot to drill a hole in your head, and that's built by a company, founded by a fellow who specializes in crypto and transportation. You decide, but you
These are the
can.
choices, crypto.
These would be some choices. So in our case, you would be able to just peel and stick the device on your forehead in about half a second and remove it as quickly as well. So, we're interested in the asymptomatic individuals. So, for example, if you look at HIV, how many people roughly do you think get HIV in the US every year?
Well, if this were a case study and I were in an interview, I would say, well, there are 330, you say in the US, million.
In the US, roughly.
And if I were to say, what percent, I'd have to assume what percent of them are having any kind of blood transfusion and what percent of them are having perhaps unprotected intercourse and what percent of them carry it. I would say maybe it works out to 0.1%. So I'll say 3 million a year.
Okay, well, luckily it's a bit lower than that in terms of people, at least in terms of people who were diagnosed. It's possible the number of people who get it is much higher. But if people are diagnosed in the US, roughly it's about 50,000 or so. But to your point,
I was off by a factor of many.
But
I would still get partial credit on that interview because it showed that I was thinking.
You're thinking about it, absolutely. So, but okay, so then extra credit here.
Somebody just wrote no credit, and John wrote that the former CEO was Ethor Bender.
That's right. And the Ethel Bennett is correct. How many HIV tests do you think are conducted roughly every year in the US?
I would say a fraction of those because most people don't. I don't know. I would not get credit for that one.
Actually, it's about 10 million. So what's interesting here, right, is that knowing that you don't have HIV, that's very important, And so people think about certain markets like Parkinson's.
It'd basically be anyone who takes an STD test.
There you go, exactly. Right. And so you can look at Parkinson's and say, for example, this is a very small number of people who get Parkinson's every year. It's about also 50,000 or so. But actually, knowing you don't have it and are not going to have it, that's very important. So if you have a technology that can screen your brain in real time for diseases that you might be developing, but for which it will take years for you to become symptomatic, you want to know this early, especially if it's brain cancer, for example. Mm-hmm. When it comes to Parkinson's, there are all kinds of diets that appear to be neuroprotective. There was this fellow in Germany, Dr. Brack, who came up with a very, very interesting hypothesis. When you speak to neuropathologists, they usually brag about how they can make thin slices of the brain. It's as if they're working on some sort of carpaccio or vitello tonnato. But actually, this fellow had a different idea. He thought, I'm going to take thick slabs of brain tissue, and he did that because he was interested in monitoring, the alpha-synuclein clumps in the brain, sort of Lewy bodies. And he found that, in fact, it seemed that they were moving from the back of the head to the front of the head, And then he posited that perhaps Parkinson's started in the gut. And so there was an experiment that was done on mice where they injected misfolded alpha-synuclein in the gut, and a number of them severed the vagal nerve. And lo and behold, none of the ones with the severed vagal nerve had any clumps of alpha-synuclein in their brain and had any type of, motor symptoms that you would sort of see in Parkinson's patients. So again, if you were to find out that you are at risk for Parkinson's, you might, for example, change your diet, because now there's more research that's been done on the effect of diet on Parkinson's. And we also know, for example, in terms of dementia, that it's not a very good idea, for example, to take Benadryl or these types of cholinergic affecting drugs. So we're moving into a world where we're going to be screened for risk factors, not necessarily just in terms of the genetics, but also the physiology. And you will have the ability to adapt to that before you're sick. So we're building sort of the tank lights for the brain and telling you, that you might have a problem later down the road. And if we think you're going to have brain cancer, you would want to know this when a tumor might still be small and operable, rather than, a couple of years down the line when you wake up and all of a sudden you don't see anything in the left or right hemisphere. If we think that you have Parkinson's, you might be developing Parkinson's, you would want to know that early as well. If we think you might be having an adverse reaction to a drug, you would want to know that early. And then, of course, the other case is people who are paralyzed with, from a stroke or ALS, clearly enabling them to communicate and doing so non-invasively is a wonderful thing.
Before I give the floor to my friends here, I have two questions that I hope are like very short answers, just like a yes or no, because that's what my mother-in-law can understand. Number one is, what % of your work is diagnostic in nature?
100%.
100%. And you have presently no aspirations for non-diagnostic work.
Hang on. That's well. If we decide that curing locked-in syndrome with a computer and algorithms and devices is a therapeutic, if you decided this is sort of a 21st century patch, then maybe the more appropriate answer is 75%.
I personally wouldn't say that's. I would think that's more of a therapy.
Assistive technology.
Yeah, right.
Yeah. So exactly. So then if it's more of an assistive technology than a diagnostics, then you could say 75% would be, looking at the
brain. So a quarter of your effort goes towards the assisted technologies.
Yeah.
Okay, fair enough. And I'll be damned if I remember the second question. I was so blown away by the first. Luke, what were you going to say?
I don't think that the power in this system is for the individual user, that you can actually identify all these diseases on an individual basis, because there are so many parameters that can go wrong, but that the actual power of the system is in that you can measure a complete population, and that over a complete population you recognize some factors. Like
that's partially true. So it's partially true. So
okay.
The fact that we can have a device that's smaller than your car key rather than that Medusa you're wearing right now, means that we can actually put this on, very vast numbers of people, and we can do population analyses on those. However, if we look at the clinical trials we've done with some of the world's best pharma companies, what we have found is that very often we could see changes when individuals were put on a new drug, So looking at individuals longitudinally is very important, because, you want to be able to follow specific individuals and have a sort of a tailor-made approach. And that's extremely important. And we can do that with very few people. I'll give you an example. We did a very small study years ago with the US Navy. And so they had taken one of our devices. This was the iBrain 1. So it was bigger than this iBrain 3, still not as huge as what you're wearing, but they recorded from an individual. They sent us the data, and they said, well, what do you see? And so we said, okay, well, we don't see, any biomarkers that would suggest that this person is a candidate for, schizophrenia or for dementia, or for I think it was depression. But we see biomarkers that suggest that this person might have been over-medicated with an SSRI, and that he or she might be a candidate for PTSD, post-traumatic stress disorder, and traumatic brain injury. And within 45 minutes, the Navy responded and said, "You're right on all 6." And this was one individual, and it was one screenshot in time. That's all it was. So
is it not more like that? You see a pattern, and then you can say 80% of the people who shown this pattern went to the cardiologist and had a problem, or went to the neurologist and had this problem, but not that you can say you're 100% right each time.
I'm not saying that. What I'm saying is, if you go to a bakery and ask for an average pastry, that's not going to exist. So you can do population dynamics and population analysis if you have a biomarker that statistically is changing across people. very often you can actually get the biomarker in the individual. So monitoring a person longitudinally is important, especially if he or she has had a change in treatment, for example. So I agree with your first point, which is by having a small device, you can get population activity. But what I disagree with is the notion that that's the only thing we can do, because you want to be able to track individuals and look at how they progress. And if you can use them as a benchmark and see how they progress, many of these diseases that we're interested are neurodegenerative. So the biomarkers are going to get more expressed or reinforced over time. And that's something you want to track.
Let me make an
use AI for that?
Luke, let me
Sorry.
administratively ask, do we lose you in 8 minutes, Philip?
I can go for another 10 after that. I've got a call, I think at 9:15, so I can go a little longer.
I appreciate
It's
that
a
very
small
much.
follow-up question. It's about, do you use AI to investigate all those signals? And aren't you afraid of any bias on your on the group you're investigating in that?
So I use unsupervised algorithms.
You use what?
Unsupervised algorithms.
Okay.
So
Explain.
you're asking about AI and biases. So why don't you explain what you consider, AI, and then I'll tell you the difference between that and what you're talking about.
Okay, there's this guy at the university, local university here, who has about 6,000 hours of investigation of these kind of gaps, and he's interested in all kind of brain diseases. And they compared the ones that they considered not ill to the people who were ill. And then they did a study afterwards from the people who were not considered ill and still developed this ailment. And then they used AI to see what difference was in those two groups. But then the group they used it on were middle-aged women mostly. So you have a bias towards white middle-aged women. So how do you try to get by this bias?
Which is why you're just defeating your first point. You're just
defeating my first point.
Yes,
What?
because okay, it's two things. Number one,
and I'm going to have to limit you, Luke, after his
but
answer.
this is important.
Okay.
These are very good points. So you're referring to supervised learning here. So you've got a training set, a testing set, and you don't have an extrinsic learning rule, right? So use the AI, and everything is sort of embedded in the hidden layer. When you use a supervised algorithm, the algorithm inherits the biases, whether it is from the sampling biases or the biases of the investigator. What we like to do is we like to use unsupervised nets. So the idea is we don't necessarily program how many clusters there are going to be in the as an output. So if you look, for example, at my PhD thesis, I created a mathematical transform that enables us to see, all these different brain patterns, in a frame of reference that's created by a single EEG without forcing a priori the number of sticks. So if one were, so now I'm going to tell you how, there might be a slight paradox. So if one were to do what you suggest, which is this brute force supervised net, then, and you were to apply this to a random sample, then you're absolutely right. There could very much be a bias, which actually makes the bigger case for why we want to do longitudinal studies, right?
I get your point.
Yep. But thank you for those questions. They're great.
Okay.
Thank you, Luke. Jonathan, you had a comment about another keynote speaker I've enjoyed, Mick Farrell over at ResMed. Go ahead.
Oh yeah, wonderful.
Yeah, I was curious because I'm very much in that space of bringing medical to consumer. We did that with Cephaly, which is a neuromodulator for anti-migraines. There's brands like Think and other types of products. But I was thinking, ResMed, they came out with this brand or company called Sleep Score.
Sleep Score, Colin Lawler.
Yeah, Colin is
it used to be
together
Yakima.
years ago. And I'm really interested in hearing your thoughts about how you see mass utilization so that you do get this longitudinal perspective.
Well, so that's interesting. So Stephen Hawking used to say that with every equation he puts in the book, he makes, he halves his readership, So it's sort of similar in biotech, I think. The more complex you make a device, the less likely it is you're going to get a large population to use the technology. Now, you might end up with a lot of very detailed data from very few people, with a device that's very, involved, although you might also disrupt the individual so much that he or she might be so stressed that the data that you're recording is not, typical. Sort of the Heisenberg, uncertainty principle for biology here. So my solution is to make devices as easy to use as possible, decrease the barrier of entry, and in essence, make a consumer device in terms of scale, ease of use, and price, but a medical device in terms of rigor and precision. And I think if we can do that for the brain, then you're going to see, far more people using these types of technologies. But there is a big risk, however. You want to be very careful, because you don't want individuals to start self-diagnosing, deciding one day they have Alzheimer's and jumping out of the window and so on. I think we want to create tools that are going to empower physicians here, work very, very closely with them. And that will be helpful to them too, because they will be able to track their patients remotely. They'll be more prepared when they come in, and they'll be able to have far more data from them. So I think the idea is, one of the reasons we're very excited about Wyoming is that it is an extremely rural population. So you have about 683,000 people living there, but by land it is the 10th largest state in the country. And during the winter, a physician cannot just send a helicopter for every 911 call when the roads are completely snowed under. So what we would like to do is empower these clinics, give them these tools so that they can collect home-based biomarkers from all of these individuals. And that's why we're doing our beta over there. Wyoming is going to be to this Manhattan Project for the Brain what Los Alamos was to the original Manhattan Project, except that we don't end up with a mushroom cloud, but actually technology that will lift all the boats and empower humanity.
I just have to say that my community is so lovely, looking out for one another, that Dwayne writes, Jonathan, you really need to put a battery in your fire detector. I mean, the smoke alarm. I mean, it's not helpful that way. You want to diagnose whether a fire is coming?
I was just working on it when you guys.
Yeah, I know, exactly right. My question now for you, Philip, is, I used to work at, you may recall, Cardiac Science. We made AEDs, and cardiac arrest just can hit you out of basically nowhere. There's not a lot of like, oh, I saw this coming, and you got 10 minutes to survive. I keep coming back to this notion of the Holter monitor, or when would I be aware enough to avail myself of your technology? Would I be complaining to my GP and they send me to a neurologist? And how would I?
Every time, so the different use cases, right? So one is this in senior care homes. Every time an individual is put on a new medication, you could see this technology being applied. So you'd actually record data before the person actually begins the treatment, and then afterwards, which is exactly what we've been doing with these big pharma companies, but we'd be doing this with the senior care homes. What we found with the pharma companies is that they don't like bad news. And so if we tell them that we think that one of their compounds might have some side effects, it was not unusual back in the day to find a backdated termination letter because they were afraid to take our findings to the FDA, which was very, very suspicious to us. But if they had listened to us, they would have actually saved billions of dollars, because we tended to be right when we told them that we thought a particular compound might not be doing exactly what they thought it was doing.
I'm thinking that betting against you in general is a bad idea. I
don't know about that, but the point is we certainly looked at these data very, very carefully for years, and we give them analyses that had statistical chops to them. So the fact that we can now do this on an individual basis with a device with a smaller footprint in senior care homes, where the senior care homes are actually interested in keeping their clients healthy for as long as possible, that's a very good alignment. So you could imagine, with all of the bad press that senior care homes have received during the pandemic, that they would very much want to have access to the latest of their technologies in order to find out what is happening in their residence as early as possible. And we're seeing that kind of demand right now. So that's one area. The other one is when you go for your yearly physical, you could have your GP that says, okay, we would like to just do an at-home brain screening. Take this device and there is an app. All the data is analyzed by NeuroVigil. We will see if you change over time or if there are any interesting biomarkers there. That's where we actually.
I wouldn't typically cut you off. I just know that our time is precious. I want to ask. I read on your site that your valuation is remarkable, and is that because?
I don't think so. I don't think it's remarkable.
Okay, well, for the folks who didn't read what I read, the perspective is that it's twice the combined valuation of Google and Facebook, I think I read.
So the seed round was twice Google's and Facebook's seed valuations combined. The Series A was 2.5 times Google's and Facebook's seed valuations.
A very, 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 very big number. Is the concept that the addressable market is what? I mean,
the addressable market is 1 in 3 people suffers from a CNS disorder. But we find out way too late. We're working on technologies to find out sooner. So we're interested in the addressable market being the asymptomatic market.
You cut out there as a what?
So we are interested in the asymptomatic population. So
right. So you think it might become part of every general, go in for your annual checkup, and I might have that as be part of the examinations?
That's one of the reasons I'm hiring kind of people I'm hiring right now. These are people who have that kind of expertise in terms of building technologies that have changed the world. I mean, if you look at Qualcomm, for example, cell phones used to be a luxury in the 90s, and now right. We can't leave the house without them. So
is everyone you're hiring these days typically of a science or engineering background at this point? Do you have regulatory people on your team?
We have regulatory people as well. Yeah. And we have economists.
I have to acknowledge your T-shirt. Well done, branding there in your office, Dwayne. Yeah, very nice. Unmute yourself, and maybe my generous friend could even be a guest of yours at some point. But you had a comment. You had a comment about something that he said earlier.
Yeah, I would love to get that clip of Philip talking about the low bar, making it simple for product adoption. We were on this call months ago. Gunter Wessels was the speaker, and he brought a couple companies.
Speak at 10X again. Yeah.
Yeah. Well, one of the companies he brought, the technology was amazing, but way too complex. And I would be willing to bet that they would never get product adoption because of how the information was presented to the individual. So I think it's great. I mean, we need high tech products, but the way it's presented to a patient, I mean, you would exclude 80-90% of people, right? There's no way anyone's going to look at that.
On your head.
And so I just loved what you said. Yep.
How close are you to commercialization? What would you guess is a reasonable?
We've been on the market since 2009, right? So with iBrain 1, with the 3W, it's going to Wyoming, in a couple of months. So our beta is happening in a couple of months. We're doing some fine tweaking right now, but it's going to go to Wyoming for many of the purposes we've discussed, and also to Philadelphia for ALS and stroke.
How many units are you producing at this point? Like in the tens?
This is for the beta. So this is for the beta. So I'll be surprised if there were more than 1,000 units that will go out. Okay. Yeah. I'll be surprised if there were more than even 500. But there could be. But we're going first for the beta, and then we'll do all those clinical trials. We're talking to a number of sites in San Diego, in Los Angeles. There's Wyoming, there's Philadelphia, and also the UAE is very interested. I've got to go and give a keynote in neuro at the World Expo in mid-March, and you know, I have to stop by in Abu Dhabi and talk to the Cleveland Clinic over there. There's an
I have to
interesting
stop by Abu Dhabi is, I mean, if I had a nickel for every time somebody said that to me.
Well, no, but it's an hour away from Dubai, so you know.
Yeah.
And the way I drive is probably half an hour.
Who in my network might be qualified to help you? My friend Jan asks, do you have packaging engineers?
We have we could have more of those. We worked with a brilliant lady from MIT who created, you know, something which I think is really very, very special. But I would be interested to speak to more packaging engineers, sure.
Well, I'm putting in touch with Jan because she's my go-to for that. But what other disciplines do you need right now? Perhaps we collectively have in our network.
Oh, interesting. Interesting. So, you know, we are hiring a lot of no, it's true. MIT has no packaging course, but this lady was brilliant. And she was one of our mechanical engineers, and she decided to come up with something, and she did a great job. So I saw a comment that was there.
I'm just going to put Jan on the screen for a sec so you know who you're talking with when you get in touch with one another. Go ahead, please.
Very good. Well, you know, I guess, yeah, people who have experience in the consumer market, I'd love to speak to them. Branding people, you know, that would be interesting. We have a lot of software engineers. We have, you know, a lot of nice analog engineers, but if there are more, certainly happy to interview them. One of the things we're seeing lately is that Stanford is sort of becoming a feeder school. So not only in terms of Stanford students, but also faculty who are moonlighting at NV. So
yeah, they have some wonderful innovation courses or in whatever you call those facilities where they actually breed people. I've been working with a lot of them, but just make sure there's only 10 universities that actually teach packaging, and other mechanical engineers don't usually make really good packaging engineers.
I know, but this was an amazing MIT engineer, and she rose to the challenge, and in 1 month she did something which we were quite blown away by.
Oh yeah, I'm sure it can look really good, but structurally I'm not sure. So, you know, worry about magnetism and other things.
So yeah, a couple of years ago. So we
sorry, I was out of Silicon Valley, so I know there are a lot of good packaging engineers there.
Well, I'd be certainly happy to look at your recommendations. I mean, you know, the key thing when you innovate is to keep a humble attitude and learn from everybody. So, you know, if you think that you've got something we can learn from, I'd be delighted to hear you out.
I'll be sure to make that introduction, and thanks for the nice comment, Basile. I invited Nelly to come on as a. Oh, there, I have to give her special permission. I guess this is your first time I'm inviting you to speak on this network, so I had to give you a special ask to unmute. There we go.
Nelly's great to work with too, so.
I'm just kind of wondering, one of the things that I've seen time and time again, a really excellent product is sometimes they don't.
Oh, great. Okay, I had one job, and that was I was trying to give Nelly the screen, and I instead demoted her instead of. So we'll start that up again.
We have a bit more time. My assistant just moved my next call, so we've got a little bit more time if you want to go further.
I will take whatever you have to offer.
Hey, go, I'm back. You wanted to.
Okay. Thank you. Sorry, there we go. That was my bad. Technical difficulties, folks.
Hey, we all have them. Do you guys have a full quality system team where you have QMS procedures and people managing your quality management team? Have you already put that in place?
Design history files like?
Yes.
We have a couple people working on that. But again, if you have some people that you'd like to recommend, certainly happy to consider them as well.
So I actually have two companies who do quality system management. And what we do is we work to reduce your cost and quality with a virtual paper system. And so basically you can, there's two models that we have. We have one model where you can actually work with us on lease and give a small equity in the company, and we reduce your hourly rates.
I'm thinking that equity in his company is pretty highly valued. So we'll probably go with a different kind of model for that. But
I offer two models. That's what I'm just saying, is because it's like the cost of quality when you're in the innovative space is you really want to redirect it to design control, but you still need it, because during audits and during 510K submissions, if you do not have a good quality system, you're going to spend 2 years redoing your work. That's what the problem I find with a lot of quality, with a lot of your really great products, is they're ready to go, but when you go to do the gap assessment on the quality system and make sure you have your entire DHF, there's gaps. But if you bring the quality people in earlier and that they're part of the business and they're kind of working under the low key while you're doing all the great design stuff, it's very positive that way. So if you want
to inspire an idea, I'm going to invite anyone on the call to raise their hand if they feel that they have either in their network or themselves any way that they can help Dr. Lowe get to where he needs to go. So thank you for that, Nellie. And you mentioned before something about consumers and branding. I know a guy who used to work on Jell-O and Kool-Aid and Campbell's soup, and well, that was me. And I would be thrilled to keep your company more often than every 2 years. So we could talk about that as well. I might know a thing or two. That could be helpful.
That's perfect.
Let's see.
There was a question about Chicago, I saw, right?
Yeah, Elizabeth was asking about your Chicago influence, I guess.
Oh, I see. Well, it was a very interesting time because I chose Chicago. So I did my, you know, I had a strange sort of upbringing. I grew up in Paris, in the south of France and Switzerland. When I was born, my father happened to be one of the wealthiest guys in Europe. You know, if you look at the painting, you know, the La Grande Guerre by Magritte, the guy with the apple on the face.
Yes.
That was ours. I mean, Magritte made several of those, but, you know, we had one of the main ones. We had this sort of crazy lifestyle. And when I was 10 years old, we lost pretty much everything in about a week. So my father had speculated on the market.
I was 55 years old. We lost your connection there.
Oh, sorry.
I was 55. When you were 10, you lost.
So we lost pretty much everything. You know, my father had speculated on the market and lost pretty much everything. He
was this 1987? No.
There was in the late 80s, correct. And he but he really got hit in 89. And he took a gun, put it in a briefcase, and went to see his banker. And he was convinced that his banker had defrauded him. And to make a long story short, my father ended up in jail, and we went from living in a castle in the south of France to be used for 2 months out of the year at most, and a 7th-floor house that we owned that we bought from the Rothschilds in Paris, to my father being in jail, and my sister and I, and my mother living in the studio in Paris, not knowing how we're going to survive month to month. And there was a place in Switzerland.
Anyway, I have to interrupt you. Yeah, you were ridiculously fascinating before the uber wealth jail story. I mean, come on, man. Leave something for the rest of us to tell.
No, but let me tell you.
That's ridiculous.
Let me tell you what happened. Let me tell you what happened. So my father, ends up being, sentenced to prison. I go visit him there. I was very lucky because I went to a great boarding school, and they wanted to keep me there because I happened to increase the class average. So they liked having a real bona fide nerd, there. Even though I was 2 years younger than the second youngest person in my class. So we were sort of getting by, but eventually my father gets released, and he's pardoned because his attorney came up with his defense that in fact he was on halcyon, which was a drug which had been found to make a lot of people overly aggressive. And I thought at the time he just had a very good lawyer, and that was about that. It wasn't until years later, by the way, when I was doing my PhD on sleep analysis, and that I found out that, I was talking to Chris Gillen, who was one of the founders of the Journal of Neuropsychopharmacology, and I was thinking about putting a number of mice in different compounds. And Chris said, you can't use this one, it makes people completely crazy. And I realized, oh my goodness, my father was on this many years ago. And so I called them to sort of apologize that I had sort of, completely doubted the story. But it was also, yes. He's a Holocaust survivor. He hit me very late, and he's alive.
Okay, all right, you've got to stop. Now he's a Holocaust survivor as well. Yeah. I mean, give me a break.
He was in the Lvov ghetto. So out of a family of 35, 32 were murdered by the Nazis. He survived. Okay. My grandmother survived.
Who has the rights to your life story? That's what I want to know now. My son's looking to be a screenplay writer.
Well, I don't think my life is that interesting, but I'm happy to share whatever. But and then my father's uncle survived as well. He was one of the founders of laparoscopy. So he ended up being a surgeon in the Red Army Then Stalin decided to send Jewish doctors away. He became head of a hospital in Siberia. There's a food poisoning. They blamed the Jewish doctor. He was sentenced to 8 years in the Gulag. He's liberated in 48 after World War II, goes to Israel, becomes one of the heads of Rambam Hospital in Haifa, and then finds out that one of his 7 siblings, my grandmother is still alive, she didn't perish in the Holocaust. He goes to Canada, stays with my father and grandmother, and then develops the field of laparoscopy. Isaac Fogelfanger is one of the founders of the field. Anyways, going back to the story, of course, one of the ironic things is, over a decade ago, I was in Monaco with hosting a conference, and I had, the top pharma people there. And as I was running from my room to the conference hall, I noticed this little speck on the peninsula there. And I realized, my goodness, this was the house that we lost when my father had gone berserk. And it was sort of funny to sort of now be in a room with some of the top, industry people advocating for a different way of monitoring drug reactions. But to go back to Chicago, I graduated from boarding school very early, and Chicago had a tradition of taking sort of these misfits, and I thought I could fit quite well there. Another thing they were doing is, at the time, half of the courses that you had to take at Chicago had to be outside of your area of expertise. Or your area of concentration. They called it the Common Core. I think now it's about a third, and Columbia also does a third. By the time it was about half. So I thought this could be a good fit. I also liked the fact that Chandra Shekhar was there, but unfortunately he passed just when I was, just a year I decided to go there. And so what happened was, I was studying mathematics and physics, and I was forced to take a biology sequence, and I knew I'd sign up for this stuff, right? So I took a course, and I found it to be fascinating. After 2 years of working on things that were, either abstract or lifeless, it was sort of fun to study life. But I approached it in a way a mathematician would, trying to derive things and so on. And it really exasperated some of my teachers. So I remember at the end of the sequence that I took, one summer in Chicago, this lady who was teaching the course said, "Philip, you're a nice kid." I looked like I was 12 years old, So she said, "You're a nice kid."
You have a very youthful glow, sir.
Well, maybe that's the vegan thing. But anyways, at the time, I really looked like I was 12. So she says, "You did well. Now you can go back to mathematics and sort of nerd out over there. We don't need mathematicians in biology." So everything changed because there was a clerical error. Somebody, I guess, had forgotten to remove my name from a mailing list after I taken the sequence. And so Harvard was looking for one student that they were going to take for the summer. And I had already written up a whole theory about how certain tumors might become, cancerous. And I was trying to just predict it mathematically. And so I thought, well, I've got nothing to lose. I just sent this thing over to Harvard. And a few days later, my phone, there was a voicemail on my phone, and this lady left a rather cryptic message, or at least it seemed cryptic to me. So I called my friend Jawad, who was from Algeria, and I put the phone, on the answering machine, and I tell him, "Jawad, what is this person saying?" And so he was very funny. He said, "Philip, you're going to receive a lot of calls like this in your life, and you need to call back and say, 'Yes, ma'am, I'll be there.'" I said, "Are they giving me the internship?" And he says, "Yes, they're giving me the internship." So I ended up going to Harvard to test some of my ideas. What was quite funny is on the way there, a teacher who thought I didn't belong to biology because I was a math guy. And she said, "What are you doing for this summer?" And I said, "Well, I guess some people are interested in my ideas at Harvard." And she pretty much, spat out her coffee and was quite offended that it was the math geek who ended up getting the internship. But there, what was amazing was that I got to work in the lab, and I enjoyed it very much. And I was around people who were, wonderful to work with. But even then, I found it was weird for me to be teaching basic math to the lab tech. So I found out that there was this new field called computational neuroscience, which was attracting, mathematicians and physicists and computer scientists to look at the brain. So when I went back to Chicago, I took some graduate courses in that field. I was immediately at home there. So I end up being torn between, cancer research and computational neuroscience, even though there was no identity problem with computational neuroscience. So I wrote a letter to the only person I could think of who knew a little bit about molecular biology, because he had discovered DNA with Jim Watson, and he knew a little bit about neuroscience because he was going after consciousness and had a whole, field of a program of investigation at the Salk Institute. That was Francis Crick. So I just wrote him this letter. In fact, it was quite funny because I was throwing a party in my apartment in Chicago, and I didn't show up at the party. I was in the computer lab writing a letter to Francis Crick, and I sent it by registered mail. And I thought, at least I'll know if he got the letter or not. So a week later, I received something in the mail, and it wasn't even Francis' signature. It was the Salk shipping department. So I thought, I'll never hear from this guy ever. And then a week after that, I received a letter from Crick saying, you absolutely have to do neuroscience and you have to come to the Salk Institute and you have to work with Terry Sinowski. So I worked for a year first in Chicago on finches because I thought if I ever wanted to study an alien brain, why not start with a bird? Because the architecture is completely different. It'll be fun to find out what was actually, maintained between mammals and birds. What is preserved? And so I invented a new neurosurgical technique to get EEGs from these birds. And MIT and Caltech had found out that, they had made experiments on these birds and they thought that these birds didn't have complex sleep patterns. And I knew that they were wrong because when I put infrared security cameras on the birds and they appeared to be sleeping, I could see times when their eyes would be darting left and right. So that seemed that they would be in REM sleep. But nobody could get the EEG, to change. So they couldn't see this change in the EEG. And many of them didn't see that period of time because birds are very photosensitive. So if you put lights on, they will immediately wake up. And they're also very sensitive to melatonin. Some of these implants that were built were very heavy and these birds wouldn't fall asleep. So what some of these teams at MIT and Caltech did, one of them in particular, is they drugged these birds with melatonin, which expands story of sleep, and they would wake them up at the same time with light. So there was no way they would see REM. So when I realized that basically MIT and Caltech had failed because of that, and because of the fact that birds don't have a neocortex, so you cannot just put your electrodes anywhere on the brain and expect to see, the type of signal you would see, for example, on a mouse or in a rat. I thought I have to build a new system where the electrodes would be very close one from the other on a particular brain structure. And so what I did is I anesthetized these birds, and as I changed the degree of the anesthetic, and I would vary the position of the electrodes, and then I found a signal where there was this beautiful variability. And lo and behold, when I built a chronic implant, when the eyes would move left and right, you would actually see that the EEG was changing. So when the New York Times finally interviewed me about this and they said, how did you discover this? I said, it was all like California real estate, location, location, location. So anyways, that work convinced me that I could go to the Salk Institute, do my PhD, and I decided to come up with an algorithm to analyze brain patterns, not only in animals, but also in humans, and to look at the data in sort of an unsupervised way, so that I wouldn't force mammalian biases on the avian brain. And because I was able to do this on a single channel, it was sort of fun to see that happen in humans. And that then also led to the Cambridge Declaration. So Francis,
I'm going to stop you because it's just 2 minutes to the bottom of the hour, and I need the last 90 seconds to tell you the following.
Go for it.
When I got my MBA at Wharton, and when I go to a reunion, it's a euphoric experience for me, not only because I love the people, I was unsurprisingly very social there, but just the level of discourse. And I remember the only other time I felt it, like in the last decade or so, was the first time I went to Daniel's then Future Med, and now medicine. And I called my wife and I was like, this is like, I'm having that same feeling today with you. I appreciate it so deeply that the idea of me inviting you to come speak for a half hour at my event seems so paltry that I would like you to consider having a Dr. Lowe conference that I would help you do. No, seriously, you're laughing, but I could easily see hundreds of people flocking to an event that you are heading, you wouldn't do any of the, you would just be there, and your network of people, it would just beyond fascinating.
We did one in Cambridge in 2012, and I might do another one this year, depending on what happens with COVID, but it would be on the future of neurotechnology.
Well, you have my number. We've exchanged a few emails. We need to talk more. I'm on behalf of everyone on the call, on behalf of people who will watch this broadcast later, profound thanks. I really hope you have an excuse to be in San Diego on May 10 and 11, at least the 12th.
We'll let you know.
And
thank you so much for your time, for what you do for the field, for this wonderful, collegial spirit embodied by you and the people you choose to surround yourself with. This was a very productive conversation. I learned myself a lot as well, and I look forward to continuing the dialogue. Thank you so much.
Sure to let me know all the people that you need so I can help get my friends to hang out with you. Because
thank you so much, Joe. Real pleasure.
Thank you, Dr. Lowe. We'll see you all next week. Thank you, Phil.
Bye. Thank you.